35 million people worldwide are living with HIV. A new imaging technology could help fight this terrible disease. More details next.
This is Light Matters for October 22nd, 2014. I'm James Lowe. On this week's show, energy loss enhances optical system performance, magnetic nanoparticles could aid bioimaging, and a group of innovators received the Edmund Optics 2014 Education Awards. But first, here's Laura Marshall with our top story on HIV imaging.
Thanks, James. The Ebola virus is all over the news, but HIV remains a worldwide epidemic. Now scientists are opening new possibilities for fighting the disease by studying the motion of HIV proteins on the surface of the virus. A team at Weill Cornell Medical College used single-molecule fluorescence resonance energy transfer imaging, or SM-FRET, to get a real-time view of how those proteins attack and infect human immune cells. In the first of two related studies published in Science, this new technology was used to look specifically at the processes happening with the HIV particles. Fluorophores were inserted into the virus's outer covering, called the HIV-1 envelope, which mediates viral entry into host cells. The SM-FRET imaging allowed the researchers to visualize how the molecules moved over time as the virus proteins changed position. The researchers found that in the presence of synthetic CD4 receptor proteins, GP41 subunit proteins, which play a key role in infection, were exposed. And antibodies known to exhibit some effectiveness acted to prevent a GP120 protein from opening, prompting a decrease in the virus's ability to infect cells. The related study, published in Nature and led by the National Institute of Allergy and Infectious Diseases, used X-ray crystallography to capture a 3D structure of one of the protein conformations discovered in the previous study. Both imaging techniques could help scientists describe the function of molecules from the perspective of motion. Data obtained in the studies could be used to screen the impact of drugs and antibodies that can shut HIV down.
It sounds paradoxical, but increasing the energy loss in a laser system could actually enhance its energy, performance, and efficiency. A team from Washington University in St. Louis has discovered a way to reverse or potentially eliminate energy loss in a laser system by adding to that loss to increase light intensity. Through several experiments, two tiny, directly coupled silica microtoroid resonators were used, each joined to a different fiber taper coupler that aids in guiding light from a laser diode to photodetectors. In estimating the intensity of the light in the two microresonators, the researchers found an initial decrease in the total intensity of the resonators, followed by an increase and subsequent rebirth of strong light intensity as the energy loss was increased. The researchers have also achieved two nonlinear phenomena: the thermal effect caused by redistribution of internal energy in the system, and a Raman gain in silica despite increasing loss. These findings could lead to new techniques for controlling and reversing the effect of loss in various other physical systems, including photonic crystals, plasmonic structures, and metamaterials. The research is published in Science.
Nanoparticles that fluoresce to enable tracking in the body can now be precisely manipulated with magnetic fields, according to researchers at MIT. Manipulating the particles with electromagnets is key to using them in biological research, as the tiny particles could otherwise get lost in the jumble of molecules circulating within a cell. A new technique developed by the MIT researchers involves magnetic nanoparticles that cluster together while fluorescent quantum dots form a uniform coating around them. These super nanoparticles can also be coated with silica to seek out and bind to particular molecules, such as markers for tumor cells or other disease agents. Other research groups have achieved different combinations of fluorescence and magnetism, but a nanomaterial that incorporates both has long evaded scientists. Typically, they have been too large to make practical probes of living tissue. Future experiments could involve attaching additional materials to the particle's coating so that they interact in specific ways with molecules or structures within the cell, either for diagnosis or treatment. The research was published in Nature Communications.
With projects ranging from fluorescence-guided cancer surgery to water quality assessment, 9 graduate and undergraduate optics programs from a pool of nearly 1,000 will receive Edmund Optics 2014 Education Awards. The winners represent universities around the world, from the United States, Europe, and Asia. Each will receive Edmund products. For more information about those innovators and their work, visit photonics.com.
That's it for this week's show. Email us your questions or comments at lightmatters@photonics.com. And don't forget to follow us on Facebook and Twitter for news updates every day.
Thanks for joining us. We'll see you next week.